All right.
Very well. My name is Mario Grijalva, and I am the project director on the, or co-director along with Martin Llewellyn of the VECON-VEN research network for vector-borne diseases surveillance and control in Venezuela and the Andean region. Venezuela has been in a continuous downward spiral over the last decade or so. And this has translated into erosion of the scientific capacity of the country and the ability to control diseases, which has an effect in the whole region, and it also has a deep effect in the population of the country. So this network is a response to this situation, and I want to welcome everyone to this symposium. So without further ado, I will yield the screen to Dr. Martin Llewellyn from the University of Glasgow. Martin.
Thanks very much, Mario. That's wonderful. Yeah, so welcome everyone to this online symposium. Obviously, we thought we might get a chance to do this in person, but as things have progressed, it's become clearer that this is going to be the future of meetings for a while. So Mario's told you a little bit about the rationale for the network and why this came about. I just clicked through. So I'm just going to introduce things for a few minutes prior to the actual symposium itself beginning. So we really set up this network to try and support Venezuelan vector-borne research and regional vector-borne disease research, around Venezuela, especially in light of as Mario said, the declining and worsening situation at present. And the idea was to try and develop new and potentially interdisciplinary approaches and context-dependent approaches to try and address vector-borne disease control in this setting. So against the backdrop of worsening socioeconomic conditions, migration, etc. And one of the key goals that we think our network, where we can really get good bang for our buck, is to try and support Venezuelan researchers, senior researchers, but also especially early career researchers, and try and build research capacity or rebuild research capacity in Venezuela, as well as in the Venezuelan diaspora, for the sort of the next generation of researchers coming up. So it's really key that this regional cooperation takes place, and the sort of founder members of this network are really three universities within Venezuela. So Carabobo, Central, sorry, not a university, but an independent research organization called the Incubadora Venezolano de la Ciencia. And then we're working in cooperation with CISEAL in Ecuador, University of Rosario, and Fiocruz. So it's, as I say, it's an open network. So these are really just the founders. We're very keen to meet and get on board new national, regional collaborators, as well as obviously international collaborators in Europe and the US, potentially elsewhere. And we are really interested in trying to support data exchange, researcher exchange. We're trying to reach out to stakeholders, and we're trying to identify opportunities to fund research on vector-borne diseases in Venezuela, as well as to continue funding interactions between scientists. So this is a timeline. This is really where we are now. So we actually did have a meeting back in Glasgow in 2017, sorry. But we've been a sort of informal network for a while. We met in Ecuador in CISEAL in 2018. And then in March 2019, in light of all the sort of the information exchange that had gone on at the meeting in Ecuador, and sort of the understanding of really the terrible situation which was building in Venezuela in relation to vector-borne disease transmission, and especially the malaria outbreak, which is ongoing. We produced a couple of meta-analyses, reviews, which to try and bring attention to what was going on. That led to the kind of current project as it stands. Excuse me. And we had our first meeting for this current project, which is UKRI funded project in Colombia in 2019. And I'm very pleased that we did actually manage to squeeze in a meeting, a physical meeting before at the beginning of 2020 for the obvious reasons. And in March 2020, things changed really quite dramatically for everyone. And we as a network, VCon then were able to secure some emergency funding. And I think everyone's lives have been pretty much turned upside down. And certainly the focus of our network necessarily has shifted away from vector-borne disease control to the very important and practical task of controlling coronavirus in Venezuela and the region. And that's just a photo of Dr. Neuer there, Professor Neuer, in full PPE. So for the last year, from March 2020, sorry, that should say February 2021, we have been really concentrating our efforts on coronavirus surveillance, control, modeling, and trying to support the effort to understand what's happening with coronavirus in Venezuela. So hopefully in this meeting, we're not actually going to talk that much about coronavirus, although obviously coronavirus is the context for everything now. But we really felt that we needed to have this meeting to try and begin to get the momentum back for vector-borne disease research and surveillance in the region. So some research exchanges have already started. So there's now Venezuelan ECRs, early career researchers who have traveled to Colombia at the beginning of this year, and we've got this meeting here. So hopefully this is going to catalyze efforts to sort of get back in the driver's seat, and push our networks forwards. Obviously, in this kind of brave new world of coronavirus. So the principal aim here then of this symposium is to really sort of restart the network in some ways, and try and get our momentum back after a very successful year. But obviously we are still in the middle of a pandemic. Some people, don't know really what direction it's going to take. And Venezuela certainly looks like it's heading into its second wave. So I don't think we can ignore the context. And I think Maria Eugenia is going to be talking about that in her presentation shortly. But obviously all these vector-borne diseases that we've been researching haven't gone away. They're still there. And it's time for us to begin, well, not that we've really stopped, but certainly within the context of our network activities, begin to focus on them again. We're really lucky to have got a lot of new partners and speakers, I should say, and collaborators with fresh perspectives to come and talk to us over the next few days. So you'll hear a few talks from VCon Ven researchers, but for the most part, you're going to hear talks from people from actually outside the network. But I hope they'll join us in our network to try and get some fresh perspectives on the problems as well as the solutions, potential solutions. And we're very lucky to have a couple of funders who'll come and talk to us about funding priorities in the region as well over the next couple of days. So this is what the agenda approximately looks like. So I'm sure you're all pretty used to Zoom now. As we run through the talks, please pop your questions into the chat. our aim is to have lots of time after talks for lively debate, but we will keep an eye on things, as certainly tomorrow afternoon there's a chance to brainstorm after the session. So we'll try and have as much lively discussion as we can, as Zoom allows us, over the course of the symposium. On so at 2:30 today, we will have a closed session for the network and the speakers, just to give you just to have a general meeting and an update on where we are. And we will also, if that runs on, so for those network members, you'll see that there's a Google Doc with the meeting agenda on it for you to look at. And that's there to update you on our activities, and really try and think of a way of forging forward how we're going to do it. So that's today's agenda. Tomorrow, we've got lots of great talks on around generally the theme of diagnostics. I should say that the title of today's symposiums principally on vector-borne disease control. This Tuesdays is principally on novel diagnostics and approaches to surveillance. And like I say, we might be able to spill the meeting over into that brainstorming session. Alternatively, we can have a session on Thursday at 4 p.m. outside the symposium. That's for the VConven members. And then on the 24th, we're going to be talking about community engagement, which is super important, especially where you're trying to sort of build resilient surveillance and control infrastructure where there isn't a great deal of top-down organization. So how do we get communities to empower communities to take charge of their own vector-borne disease healthcare needs? Right, and that's about that for me. Please do, if you haven't already, if you're watching this on the streaming channel, please visit the VConven website, join the network, get onto the mailing list. If you're a laboratory scientist watching this and you're interested in knowing how you can get involved, also have a look. There's ways of donating useful equipment for ongoing VBD research in Venezuela, and do follow us on Twitter. But without further ado, I shall hand you over to María Eugenia, Professor María Eugenia Grillet from Universidad Central de Venezuela, and she'll take it from here.
Infection diseases. Okay, let's start. Sorry. Okay. Vector-borne diseases account for more than 70% of the global disease burden and around 1 million of fatalities per year. They are very common in all the epidemiological landscape in the Americas, and particularly dengue, Zika, and chikungunya are the dominant in terms of spatial distribution, followed by malaria, Chagas, and leishmaniasis. Dengue is one of the fastest growing global infection diseases. Recent modeling estimation project 400 million of infection and 40,000 fatalities per year. The global incidence of this disease has increased in the last years. And in the Americas, the transmission has spread to new areas as a result of the Aedes aegypti spreading. In this map, you can see the global variation of dengue transmission, with Venezuela showing one of the highest transmission of dengue in the continent. Here, the dengue incidence has increased along during the last 25 years along the geographical spatial distribution. The current status of Aedes aegypti and Aedes albopictus is unknown because in the country since 2016, entomological surveillance and reporting has been suspended. Dengue is in the global agenda for control and elimination of vector-borne diseases. The specific targets for this disease are for 2023, 2025, and 2030, where it's expected that 0% of case fatality rate due to dengue. The frontline intervention of dengue, the main intervention for dengue is vector control, and for sure our next speaker is going to talk about that. Malaria remains as a major cause of death and illness in the world, with more than 200 million of cases and 400,000 fatalities per year. The control and eventual elimination of malaria is one of the greatest challenges in public health, so now competing with COVID. In the America, the malaria is still an important public health, especially the malaria by vivax, where 4 countries, Brazil, Colombia, Peru, and Venezuela, account for more than 80% of the cases. But in the last year, Venezuela is accounting for more than 50% of these cases, and currently Venezuela is considered the main hotspot of malaria by Plasmodium vivax, as you can see here, and Plasmodium falciparum. The global technical strategy for malaria for 2030 is consists in reducing malaria incidence by at least 90%, reducing malaria mortality by at least 90%, and eliminate the malaria in at least 35 countries. Early diagnosis and vector control are the main frontline intervention, particularly the use of bed nets and the insecticide indoor residual spraying. The main challenge to malaria elimination from the point of view of vector control in the Americas is that most of the vector species are exophilic, exophagic, and early evening feeders. So for a successful vector intervention in the Americas, we need more funds for vector surveillance, and intervention. Indeed, South America is a kind of neglected region for that matter. To promote training program for vector biologists and incorporate the community in the malaria control programs, to focus intervention and planning in using modeling to develop new approaches for vector control of our vectors in the region, more accurate identification of transmission in hotspot, and routine evaluation of control strategies and insecticide resistance. Indeed, this is one of the main challenges to malaria control and elimination in the world. And we have few studies in the region addressing this aspect. We don't know, for example, what is the impact on blood feeding behavior and vectorial capacity of the insecticide resistance. And here you can see is a paper for Africa, where you can see the percentage of mortality of Anopheles gambiae and Anopheles funestus along the year. Has a way to see the effectivity of the insecticide has diminished along the year. And this is a map of the resistance to the main insecticide using in Africa. Another big challenge for malaria control and elimination in the region is the high biodiversity of vector species that we have, with different ecology and different vectorial capacities. For example, the Albitarsis complex is a very important example for this. This species complex has around 8 species, and we don't know yet how many of them are important vectors in the region, and this is particular this is a particular issue for Venezuela. We need more studies to address this aspect. Another challenge to malaria control and elimination in the region is the deforestation or the land use change. Venezuela is an example for that. Here you can see an image of the south of the country, where most of the malaria has increased in the last 20 years. These red spots are deforested areas. And we have found that changes in land cover by gold mining activity explain the long-term increase of malaria risk in Venezuela. Here you can see that in these deforested areas, you have more hotspot of malaria and more breeding site of the main vector species Anopheles darlingii and Anopheles albitarsis. So the change in the vegetation, it creates new habitat for the vector of malaria in this area. Indeed, Anopheles darlingii, which is the main and dominant malaria vector in the region, is an invasive species and good colonizer of perturbed habitat. And this is a problem in Peru and Brazil, and even including Colombia also. The political instability and socioeconomic collapse is another factor promoting an increase of vector-borne diseases, and Venezuela is another example of this. The steep drops in oil revenue and rise in illegal mining have effectively shut down many aspects of Venezuela's public health system, with a huge impact on vector-borne diseases and interruption of control activities. Here you can see the malaria cases for Venezuela from 2000 to 2017, and this is the curve of the annual inflation in the country. Indeed, illegal gold mining is one of the main local socioeconomic malaria driver in southern Venezuela. So controlling malaria in this mining setting is a big challenge in the country currently. Cross-border malaria, another challenge to malaria elimination in the region. We called the attention to that in 2019, when we showed that from 2015 to 2014 to 2016, the number of cases of imported malaria from Venezuela in the north of Brazil increased significantly. This is a recent paper, cross-border malaria in northern Brazil, where they bring more evidence of that. For example, you can see here that in 2018, Venezuela accounts for around 80% of the imported cases in the north of Brazil. Indeed, the regional crisis of the departure of Venezuela can affect or is affecting the countries in terms of global strategy for malaria control and elimination. The potential risks are reintroduction of the disease in new areas, even where it had previously been reduced or eliminated, as is the case of, for example, Argentina or Paraguay. Two countries certify with the elimination, and enhanced malaria transmission in both borders, as you can see in Colombia and Brazil, and promote the spillover of cases, and you can see in the whole region, and even the spread of drug resistance, not yet documented. Currently, in 2019, the country again reported around 400,000 cases of malaria. These represent the 53% of the cases in the region, and 73% of the total fatality from malaria in the region. But World Bank is still labeling Venezuela as an upper middle income country, even though the economic situation has changed and the poverty has increased. Venezuela needs to be prioritized by key funders for research and control. And finally, our last new challenge to malaria and vector-borne disease control and elimination, COVID-19. This for sure is affecting many disease control programs, including vector-borne diseases. Okay, thanks.
Okay, can you see it?
Yes.
Okay, so the idea
yes.
is to talk about surveillance and control of Aedes aegypti, and I'm gonna just do a quick mention of the tools, and then I'm gonna try to answer some questions about why control is not so effective, and some requirements for effective vector control or Aedes aegypti control. And how do we know that we actually achieve effective control? And try to speak a bit about our work on the question of how many aegypti can cause epidemics. So this is a list of the Aedes control approaches, from the current approaches to the newer ones. And on top, you can see the strategy that I call seek and control, because we move where we think mosquitoes are. And so we apply control there. And that includes source reduction, that is container elimination or modification, environmental sanitation, the application of larvicides once we find the containers, and then the use of insecticides to control the adult mosquitoes. Then there is this strategy of lure and control, which means trapping mosquitoes, attracting mosquitoes to traps or trap devices. And then newer auto-dissemination approaches, in which the own mosquitoes are used to disseminate the control agent. So I'm gonna just go right quickly through these methods that I guess that you are all familiar with. So the control of immatures involves environmental sanitation or transformation, the elimination of containers, improving public services that generate containers like water storage containers, trash. And then it's important to include the communities in health education. We then have ways to directly eliminate the immatures using herbicides. We've been producing these formulations of bleach, domestic bleach. And then there's a number of larvicides that are very effective. The most commonly used was Temefos, which is an organophosphate insecticide, but there is resistance everywhere. But the other biorational larvicides are very effective, although they don't last long or long enough. Then we have biological control agents that face the problem that many of these are small containers, and so we need to seed them again and again, but they can be quite effective. Then there is a classical use of insecticides against adults, mainly using an ultra low volume insecticide that is highly concentrated. And this is not a good tool to manage Aedes aegypti populations, but to eliminate infected adults somehow. It hasn't been very effective. You can use ULV indoors, or we can use residual insecticides indoors and outdoors, although that hasn't been used that much for Aedes aegypti. Then we can use barrier applications to intercept mosquitoes outdoors and vegetation, etc. But the main problem is that there is a generalized resistance to most insecticides because of the reliance on them. Then there is a new, well, not so new, but this devices, such as curtains, covers, screens impregnated with residual insecticides, and some ovi traps with insecticides. And then here we have a trap that we invented a few years ago. And this trap is a 20-liter bucket that has 10 liters of water and a hay pack. So it decomposes and attracts rabid females of Aedes aegypti and Culex, Quinquefasciatus. And mosquitoes are trapped in this special insect glue. So it doesn't use insecticides. And we can use this trap. We've been using this trap for surveillance and for control. And I'm going to talk a little bit more about it. And then the personal protection measures, protective clothing, and then the spatial repellents that can help somehow. Now, among the auto-dissemination devices, this is a container with water that attracts mosquitoes, and they get contaminated with an insect growth regulator called pyroproxyfen. So the idea is that the female gets contaminated, so when she goes and finds another aquatic habitat to lay eggs, the pyroproxyfen gets into the water to suppress the emergence of the pupae. And we have the genetically modified mosquitoes, mainly from a company called Oxitec, and they release males that carry this dominant lethal gene, and they mate with the wild females, and then the lethal gene expresses in the advanced 4-instar larva, and then it dies. It's been tested mainly in Brazil currently. And then we have the approaches of using Wolbachia bacteria. These bacteria are endosymbionts of many insects. It wasn't present in Aedes aegypti, but it was transfected, and then now it's been used for two purposes. One is population replacement that involves releasing male and females Aedes aegypti infected with the bacteria, with a strain of Wolbachia that blocks the dissemination of virus within the mosquito and therefore reduces the possibility of transmission. It's been tested in many countries, apparently with good results, reducing the dengue cases. And the other approach is population suppression. In this case, only males carrying a Wolbachia bacterium are released. So when they mate with a wild female, there is cytoplasmic incompatibility, meaning that the eggs are not viable. So that's the main two uses of the Wolbachia approach. So one of the questions is why Aedes aegypti control is not so effective. We have the tools, or at least some are emerging, that have potential. And what happens is that Aedes aegypti is a species that does extremely well and transmit arboviruses at very low population densities. So one question we're interested in is what is the density, the minimum density of Aedes aegypti that can prevent outbreaks of dengue and other arboviruses? So one of the main problems is that the eggs of Aedes aegypti can last for months in the containers. And there's no specific tools against eggs. And this capacity is what gives Aedes aegypti the possibility of recover after severe disturbances such as vector control, droughts, storms, etc. On the other hand, larvicides, even though they are very effective, they don't last long. So you have to reapply too frequently, and some of these larvicides are expensive. So it hasn't been, it helps, but it's difficult to manage. Also, we have learned in the last few years that mosquitoes, Aedes aegypti is using, and also Albopictus, cryptic aquatic habitats that are very difficult to locate and treat, and they can be super productive in some cases. And traditionally, the control of Aedes aegypti has been done using insecticides against the adult stages. That has a limited effect and hasn't been very effective, especially because of the evolution of resistance that is widespread. So these are some of the factors. Of course, there are many sociological, economic factors that I'm not listing in here. It's just about biology. So I listed here a number of possibly requirements for effective and sustained control. The first thing is that we need to validate what we use. And for that, we need some experimental method and an efficient mosquito surveillance system to evaluate the impact of control, and then do this in uniform geographic areas, because we know that the ecology of Aedes aegypti varies with location. So it's going to be difficult to get one solution to all cases. And that's something that's probably not understood, because we're always looking for a universal way to solve the problem everywhere. Now, we also, of course, need to use an efficient vector control tool. It could be anything: insecticides, traps, source reduction, predators, modify mosquitoes. But if the control agent is not efficient in itself, like, for example, if there is insecticide resistance, then you know that you are losing the fight at the outstart. So you need an efficient vector control tool. Then you have to apply it with appropriate dose in the field. Use an efficient mechanism for the application of the control agent, depending on the control agent that you're using. That's something that is overlooked in many cases. Even people applying USV insecticide never calibrate the machines. So you need to reach a very high level of coverage with your control agent to affect most of the local population, over 80% mortality or reduction in fertility, and cannot leave areas untreated or undertreated. Then you have to reapply the control agent at appropriate intervals, involve the communities, and use control tools that are accepted by the communities, and whenever possible, use an integrated vector control approach. So how do we know that we actually control Aedes aegypti? Okay, well, we look at disease prevention. If there's no disease, we may claim that we are successful. But in many occasions, that's not the case. It's simply that the virus is not present. And so the only way to do this is through experimentation, perhaps using a cluster randomized trials, where we compare intervention versus non-intervention clusters, and with epidemiological human disease outcomes. Now, these trials are very expensive and rely on having these set up out there at a time when the arbovirus invades the study area, which may or may not happen. So the trial may last several years to make it even more expensive. So what if we can use proxy variables, that is, indirect indicators of transmission risk, without involving human samples and expensive trials? So perhaps exploring Aedes aegypti density thresholds in a way I'm going to explain, and following the arboviruses in mosquitoes, we might find a way to do this easier. So the definition of threshold in theory is that the introduction of a few infectious individuals into a population of susceptible people will not cause an epidemic unless the density of susceptible hosts or vectors is above a certain critical level. So that's what we call a threshold for mosquito density. So we are interested in finding that threshold in Aedes aegypti as the main vector of dengue, chikungunya, Zika, and urban yellow fever. So the advantage of knowing what a threshold is that vector control programs can have a very well-defined target. So they know when they are being effective or not. And there are several difficulties though, because thresholds seem to be moving targets, and they depend on many factors, and also depend on the tool, the surveillance tool. And surveillance is a very important component of control, and that I think that's a concept that is not popular in many places. But if we don't have a good surveillance system, good surveillance tool, it's very difficult to determine if we are actually doing a good job controlling the mosquito. Now, Dana Fox produced a model a few years ago using PUP per person as an indication of mosquito density. And he showed with this model that thresholds change. For example, higher temperatures, you need fewer mosquitoes to cause an outbreak. Also, if you have herd immunity, then you need more mosquitoes to cause an outbreak. So that's why I say that thresholds are moving targets, because temperature changes and herd immunity also varies with location. But you know, we've been trying to work on that aspect. In Puerto Rico, we've been conducting vector surveillance and control using mass trapping with this AGO trap. And for surveillance, we place between 30 and 40 traps per community. We inspect them every week to collect adult mosquitoes, and then make mosquito pools for virus identification. Now, for control, we use 3 traps per house in most of the houses. The good thing about the traps is that you don't need to service the traps, but every 2 to 3 months. That gives you some time. So this is a piece of study that we've done for 8 years, following a few communities. So what I'm showing here is 2 communities. The one on the left is with mass trapping of mosquitoes, and the one on the right without vector control in 2014, when chikungunya virus invaded Puerto Rico. And at the bottom are the same 2 communities in 2016, when Zika virus invaded Puerto Rico. So the empty bars is rainfall. The continuous line is the number of Aedes aegypti females per trap per week, every week. And the dark bars is the presence of the virus in the mosquitoes. So in 2014, you can see this aggregation of temporal aggregation of chikungunya virus in Aedes aegypti. And so in 2016, we see the same thing for Zika virus. Now, because Aedes aegypti doesn't fly very far, what happens is that this presence of virus in the mosquitoes in a continuous way for several consecutive weeks, that's revealing a local outbreak among people. So that's why I was saying before that we might be able to track transmission without having to take human samples. So this is another way to look at the data. The open circles is the mosquitoes in the non-intervention, and the dark dots is the mosquitoes in the intervention site with mass trapping. And as you can see, it will seem like if you had less than 3 female Aedes aegypti per trap per week, you may be able to prevent transmission. Note that we did find some positive mosquitoes in the mass trapping sites, but no in the same magnitude and frequency as in the non-intervention sites. And we have serological data for 2015 that showed a significant impact of protection of the local residents against chikungunya. So the advantage of the use of thresholds for these type of demonstrations, field demonstrations of vector control, how vector control impacts epidemiological outcomes, is that even though the gold standard is to do cluster randomized designs, usually with 2 arms, 1 control and 1 treatment, they're very expensive. Especially, it's difficult to get human samples, and the clusters need to be at least 20 clusters on each arm in order to have the power to detect significant impacts. However, if we know that there is a density threshold that prevents transmission, we don't even need to use controls for the field trial, because the only question that we need to answer is whether the density of Aedes aegypti is above or below the threshold. So currently, we are conducting a cluster randomized trial to determine the threshold density for dengue virus transmission. What happened in 2014 and 16, when we had this surveillance system in place and mass trapping, was that the human population in Puerto Rico didn't have antibodies against chikungunya or the Zika virus, whereas dengue has been circulating in Puerto Rico for decades. So our hypothesis is that the threshold for dengue transmission is going to be higher than 3 females, because a percentage of the population already have antibodies against dengue. So we are conducting this trial right now. We started 1 month ago. We have 1 non-intervention arm and 3 intervention arms. And those are the following. We place 1 trap per house, or 2 traps per house, or 3 traps per house in neighborhoods. And the idea is to produce different levels of Aedes aegypti in time, and then see in which of these arms we have suppression of dengue. And so that this project is to be out there for 3 years, and we hope that we're going to be able to better define what is the threshold for dengue transmission. And so that's, I think that's about all of my talk. And thanks again for the invitation. And if there are questions.